just scratching the surface of the immunotherapy in Parkinson. The first two monoclonal antibodies that were under development did not meet the primary endpoint in their first phase 2 study. However, these were small and short studies. Now, with the Prasinezumab, with the potential signal we found with the secondary endpoint, we are running a much larger study to assess whether with the longer follow-up in a more broad population, this drug is able to slow the motor progression rather than the full global progression of Parkinson's disease. This is unique because Prasinezumab binds aggregated form of synuclein, but also other species, from monomeric to potentially some species that we don't know yet, that could be the real trigger of the progression of the disease. And our study, the PASADENA study, was positioned including both treatment-naive and MAOB inhibitors, people treated with MAOB inhibitors. And the study we are running right now, called PADOVA, is testing Prasinezumab on top of stable symptomatic therapy, so MAOB inhibitors, but also people treated with levodopa. So it would be very important to assess the long-term follow-up of this drug before we can define whether or not immunotherapy is relevant for Parkinson's disease. But most importantly, we need to assess whether the monoclonal antibody that target the right pathogenic species would be the one to be successful over time. And there are many other monoclonal antibody under development from different companies. So only with clinical data, with long follow-up, we will be able to assess whether or not they are successful, because each of them is specific and they have their own characteristic.